All right, let's get going. All right, exam on Friday. I'll get up here. Hopefully I'll be able to get it. I'll be up here. So when the class gets out, is there a class ahead of us?
No.
I don't think so. But if you want to start about 10 minutes early, you can. So I'll try to get up here. We do have a class after us, so we can't dawdle. Lab this week, it is basically review for the practical that we'll have on Monday. Check your calendar is what we'll do on Monday. What we'll do later today, we'll do two things. One, for those people that are presenting the nematodes, we have to pick your date because we have 2 weeks basically to present. So look at your dates on when you want to present the nematode. And then number two, our lab practical, we'll do half the class at 2 o'clock and half at 3 o'clock, or about half. All right, so let's see. We'll take kind of figure out when everyone's coming. That'll reduce the number of people in the lab. So that way you don't like get like a waiting or a line at any one station on the practical. All right, so we'll do half and half, and that's all we do on the practical day. So you'll come and take it, and you'll be done. So 2 o'clock or 3 o'clock. All right, this is where we left off. So Echinococcus, smallest adult tapeworm, rarely exceeds 5 millimeters in length. As we said, it's basically 3 proglottids. So you have your scolex in your neck, then you have an immature proglottid, a mature proglottid, and then a gravid proglottid. Our specimens are overstained, so it's kind of hard to see some of the finer detail in mature proglottids. So while the adults are small, the larval stage is one of the biggest ones that we'll find. All right, and because of their size, they can cause some pretty significant morbidity. We'll talk about that. So there's really 2 species of concern for us: Echinococcus granulosus and Echinococcus multilocularis. Humans can get them both. All right. Granulosus is basically global. Multilocularis is more northern type of distribution. Our life cycles are similar for both, with the exception of their hosts. So canid predators are going to be the host for both. All right, but based on where they're at, where multilocularis is, primarily it's going to be foxes. Occasionally you'll get it into coyotes. For granulosus, it's basically any canine, and some felines can also serve as a host. Because of the difference in this definitive host, it kind of changes what the intermediate host will be. So if you're only going into foxes, or if you're primarily going into foxes, your intermediate host is going to be the prey item for those foxes, which are usually rodents. But if you're going into, let's say, wolves, all right, now you can start to go into larger animals because your wolf packs are going to take it down. So for granulosus, primarily, usually the larger herbivorous mammals. So let us go through the life cycle. This is also a fairly simple life cycle. I guess that's good. We are showcasing what we have. All right, so this will just be Echinococcus, just like Tinea. So just like Tinea, check out the actual hosts that we can use in our presentation. So we're going to be definitive host, which is basically usually a canine. Our adult will be in the small intestine. In the small intestine. And then you have your gravid proglotid. Will detach, so this is apolysis. All right. The proglotid will rupture and releasing the egg, and then in the feces, that's going to have our egg. So we detach, and in Taenia, that proglotid leaves the host in the feces. In Echinococcus, that does not happen. The rupturing actually occurs on its way out of the host, so that it's the egg that comes out in the feces. The egg then is going to be accidentally consumed by our intermediate host, generically a mammal, where the oncosphere will emerge out of the egg from the gut, penetrate out of the gut into various tissues, where it will transform into a hydatid cyst. Where it goes kind of produces your the pathology that we'll see. The hydatid cyst develops, has our protoscolices in there. That individual is then consumed. Where protoscolices vaginate in our host and will start producing eggs in about 2 months. And this guy only lives about 5 to 20 months. So say a year. All right, we do have some ranges. So if we were to just kind of go along here, right, here's our host, here's our environment, just for kind of clarification on where things are. Now we say we travel to the tissues. For granulosis, it often goes to the liver and the lungs. Seem to be the most common sites. For multilocularis, it seems like the preferred site is the liver. But again, it can travel elsewhere. And it's actually this hydatid cyst that is the adaptation to transmit, to increase transmission. And we'll talk about it. And it does that, it basically does that through pathology, which we'll talk about. All right. Questions? So hydatid cyst is our generic larval stage. It is a bladder worm that exhibits asexual reproduction inside of this intermediate host. So the reproduction actually occurs inside the cyst itself. So our cyst, and we have a slide of a cross-section of a cyst, and that's a very small cross-section. But the cyst has a thick non-cellular layer and then an inner germinative layer. I think the handout used ectocyst and endocyst. So that thick germinal layer is all about protecting the germinative layer on the inside. So this outer, you could say it's laminated layer, it's really thick. That's forming our protection. The host, everything else is host host immune's trying to fight it, trying to block it off. All right, but that acellular layer is just highly resistant. It's that intergerminative layer that where we see our budding, our asexual reproduction. All right, so what's going to happen is you have a small bud formed from this inner layer, and then it forms a bladder, an interior bladder, along inside of which starts to develop the protoscolices. All right, so the protoscolices themselves, that is the individual tapeworm. One protoscolex gives rise to one adult. So you have one egg, get into the host, gives rise to a hydatid cyst, but inside that hydatid cyst, we're going to have potentially millions of these protoscolices. One protoscolex gives rise to one adult. These things develop inside of a brood capsule. Right? And so you'll have your brood capsule that pinches in, forms, and then you're going to have about 10 to 30 or so protoscolices. These brood capsules are still attached to the germinative layer by a stalk. This will detach, allowing this brood capsule to drop down by gravity towards the bottom of the hydatid cyst. The collection of those brood capsules is called hydatid sand, because you've got this big cyst, fluid-filled cyst, and you see these small particles down at the bottom. That's the hydatid sand, those brood capsules. You ready? So these things, these hydatid cysts, there's actually two different types of them. And that's what, at least one thing that distinguishes between our two species of Echinococcus. All right, so all of the hydatid cysts exhibit this internal budding to produce this protoscolices inside of the cyst itself. All right, if that's all that it is, just internal budding, that is called a unilocular hydatid cyst. Internal budding only. This is exhibited by Echinococcus granulosus. So as this thing is developing, and you have more and more of this budding, more and more of this reproduction, this hydatid cyst will grow bigger and bigger and bigger and bigger. And you can imagine if you have this cyst growing bigger and bigger and bigger, you're going to start exerting pressure on the insides of the animal. You're going to be exerting pressure on various tissues inside that animal. You are adding weight to the animal. The other type is exhibited by multilocularis because it is a multilocular hydatid cyst, also called an alveolar hydatid cyst. We see the internal budding. It's producing the brood capsules with the protoscolices, but you can also have the cyst itself start to be constricted and pinch in and then fuse, producing a second part of the cyst. So internal and external budding that leads to metastasis of the initial hydatid cyst. So it is, instead of getting one giant cyst, you have a hydatid cyst that can then pinch off and produce many more of these smaller cysts, each one still having internal budding, brood capsules, and so forth. Now, not all of them will have the budding. But this is a potential then where the oncosphere starts developing. You have a splitting, an external budding, where a part can then travel to a different part of the body and start to invade other tissues. So why is that? Well, the unilocular hydatid cyst has a fairly thick acellular wall. Makes it pretty resistant to pinching. Multilocular, not so much. That allows it then to kind of come in upon itself, fuse, and then pinch off. As we said, each of these circles that pinches off still has a germinative layer. And that germinative layer can have these brood capsules develop, producing more protoscolices. So in the natural host, the size of all of these individual capsules, not brood capsules, individual parts of the cyst itself, will be similarly sized, and they're going to be fairly small. So you'll find numerous ones. But in humans, in other hosts that aren't maybe their natural host, all right, you're going to get chambers of different sizes. And they're going to pinch off, they're going to travel to different parts of the body, and that's part of our pathology. So granulosis is a unilocular. The only thing is the internal, it only grows by getting bigger. Multilocularis, it can pinch off, it can grow by expanding to various parts of the tissue. Ready for it? All right, pathology. It's called echinococcus or hydatid disease. All right. Now, this could be latent for years, meaning you don't notice it because the hydatid cyst it doesn't grow quickly. It's a slow growing form. So you might have it with you for several years and not even realize that you have this hydatid cyst. And then the types of pathology that you see really depends on the location. All right, so key thing is that these things will get bigger, especially the unilocular hydatid cyst. They'll get bigger and bigger and bigger and start to apply pressure to various tissues. Once you have that pressure, oftentimes it leads to necrosis. So you're crowding, you're applying pressure to internal tissue, it leads to necrosis. So if you have hydatid cyst that ends up in the nervous system, right, up in the brain, you'll often exhibit symptoms similar to neurocysticercosis. Similar types of patterns. If you end up in the bone marrow, which is a possibility of traveling, then you have pressure in the bone. You can cause thinning of the bone. Unexplained break could be how they deduce that it's hydatid disease. So in both of these, in nervous system and bone marrow, we're kind of confined by bones, by the skeleton. But oftentimes these things end up, and I should say probably more commonly, they end up in locations where they can grow unrestrained. Think abdominal cavity. You might think you've got, the most powerful six-pack abs around. It's nothing for a hydatid cyst. So gradually you start having this bulge start appearing. And no longer do you have this super strong six-pack. Case in point, some of these can hold up to 15 quarts of fluid. How many gallons is that? Math. Mental math. How many gallons? Almost 4, yeah. 3 3/4. How much weight is a gallon of water?
8 pounds.
About 8 pounds. So how heavy is that? What's that?
A little over 30 pounds.
Yeah, about 30 pounds. So it's like almost a bowling ball and a half. Depends on what maybe 2 bowling balls. Depends on what side. Imagine that in your abdomen. In your abdomen. I can be a little smart alecky a bit, so occasionally when my wife says, "You didn't carry a baby, 8-pound baby," I said, "You don't carry a hydatid cyst." She doesn't like that, but that's the science. But massive. Just think of this. I mean, that's in a human. that's in a human. Think of it in a small rodent, or let's say a rabbit, because a rabbit could get taken down by a coyote. All right, how much does a rabbit weigh? The big FFA fat rabbits might be what, 5 pounds, 8 pounds, 10 pounds, 15? I mean, you've seen some of those. But let's say a hare, say 3 pounds. You have one of these things developing in its abdomen and it grows up to be a pound. Wow, that's a third of its weight. How fast do you think that thing's going to be? Not very fast, which is beneficial for pack feeding canines, because now that thing's going to have, a huge number of protoscolices. That one rabbit could potentially infect the entire herd, the entire pack. All right. Now, it's going to have unrestricted, you're going to have some of the pressure associated with it, but the real danger is rupturing. Get hit, that hydatid cyst ruptures, that's a massive amount of antigen entering the body, and what ends up happening is you go into anaphylactic shock. And you really say it's almost instantaneous death. It's going to be minutes before you just black out, and that's it. That's the major harm. Major harm for this. Alveolar echinococcus, this is with multilocularis. Oftentimes you'll exhibit pain or discomfort in the upper abdominal region because it's usually going into the liver. You can exhibit weakness, weight loss, and you go to the doctor. They may suggest, hey, do you drink alcohol? Do you do some other things that might impair the liver? May actually mimic liver cancer or give the signs of cirrhosis of the liver. And then when you go get imaged, then they see the problem. They see that it is Echinococcus. So we're going to see something. What does this look like? I should probably give a warning. Hopefully you didn't eat ahead of time. Human patient. You're in the body cavity. Those are all, even the small ones, those are all individual hydatid cysts. All from a human patient. I believe this is the liver. There's another one. And you can just think, they're going to have tens of thousands of protoscolices in each of those. Now, is it going to be, are we transmitting it to a canine? I mean, or a feline? I guess if you try to make friends with the tigers that are out there, said maybe the tiger will get hungry and eat you. All right, but just, I mean, that's,
liver,
what's that?
Will the liver ever recover from this, or is this?
Yeah, the liver's somewhat regenerative, or it could. So they're not done. They're not done yet. They keep going on, but you get the idea. I can't remember how many they get. Two in this one. I mean, you'll see that. I mean, bedpans worth, 2 full bedpans worth is what pulled it out. That's probably almost 20 pounds. 20 pounds inside this individual. So we'll call it. I don't know if this will go up on the live stream or not. I've received one of those warnings. So yeah, it got flagged. I don't know if maybe because I didn't say it was, maybe I said it was kid appropriate. It got flagged because of the surgery. I don't know. All right. So yeah, that's Echinococcus. And I kind of said an important thing. Why did this thing evolve? Why did it evolve like this? Well, if you're going to go into canines kind of evolved as pack animals, pack hunters. So one individual, one deer, one elk, right, could potentially infect the entire pack. So that could explain the how that developed. All right. family, Hymenopelidae. Hymenopelidae. We covered this. Right? Yeah. So that's just going to be in our lab part. So we're not going to worry about it. We'll point out. Notice you've got your. You actually have 3 testes. So we do have some. I think we looked at tapeworms or parasites of rattlesnakes, and we found some tapeworms that had actually 3 testes in it. So, but yeah, I'm not going to ask anything about Hymenopus. Just go ahead and just kind of gloss over what was presented in lab. All right. Move forward. But we do want to talk about this. Family Dipylididae. Right, so it's going to have similar traits to Hymenopus, which is, if we went through here, it says usually 2 to 4 testes, unilateral genital board, large external seminal vesicle. Same type of deal. But usually there's more than that 2 to 4. Right? You're going to have more than 2 to 4 testes in this area. And our example that we have is Dipylidium caninum, which is our dog tapeworm. This is probably the most common tapeworm that you would encounter in our pets. All right, in our pets. D-I, kind of references the fact that we have 2 sets of male and female reproductive systems. We have 2 sets. So each proglottid has 2 sets of male, 2 sets of female. And they're going to open up on opposite sides. So on one side, we have 1 set. On one side of the proglottid, we have 1 set. On the other side, we have the second set. The other interesting thing is that the eggs, once you have fertilization, you have eggs developing in the uterus is going to break down. Right? They're going to disappear. So then you have the eggs developing in what are called egg packets. So packets with maybe a handful of eggs inside of them. And you can see these. Hold on, let's see here. I don't think we don't have gravid proglottids for Dipylidium, I don't think. But I have seen it. I wish I had taken pictures of it. But oftentimes when you have, somebody thinks their dog has tapeworms, you find them. Key characteristic is that these eggs appear as packets. Ready? I'm going to leave that up just a little bit. I'm going to go over the life cycle. So our life cycle. Where's my cursor? There it is. All right, Dipylidium. Dipylidium. Dogs and cats serve as our definitive host. Our adult will be in the small intestine. Adult is in the small intestine. Apolysis. So our proglottid detaches. It will exit the feces. Right. Now, our proglottid is very active. Right. Moves around a lot. This is where our cucumber seeds typically what you would see. And they're very active, so they don't necessarily have to come out with the feces. Sometimes they will just wiggle out on their own. Where these pergolodids then will be located near the perianal region. So still on the host, but outside of the host. These guys are very active, wiggling around. They dry out when they get out of the host. So desiccation, where they will then first releasing their egg packets. Right, so the eggs are in egg packets. These egg packets then get consumed by our intermediate host. What's our intermediate host? What do you think? What's that? A rodent. Rodent climb on your dog and cat. That's kind of our hint where we're still kind of on the animal. Fleas. Larval fleas, to be specific. So larval fleas will consume these egg packets where we have the oncosphere hatches, or the egg hatches released in the oncosphere, in our gut, or in the flea's gut, penetrates out of the gut into the hemocoel where it develops into a cysticercoid. So we are in the body cavity. Why the larval fleas? Because the larval fleas are the ones that feed on organic matter. Alright, they're not feeding on the blood of the host. They're feeding on things that live on, the organic material, which would include these egg packets. This cysticercoid then will survive metamorphosis of the host to our adult flea. Right. Where it's still cysticercoid in the hemocoel. Adult fleas now are going to bite and feed on the animal. Causes a lot of itching, quite a bit of distress. The animal's going to scratch, it's going to bite, which allows it then to accidentally consume our adult flea. Fleas. And that's our life cycle. So, what's usually found is you see some white stuff around the perianal region of, I should say, the back end of the cat or dog. And that's usually going to be your first clue. Normally you could say rice grains. I think the vet would say, well, does it look like rice grains? Because at that time you're seeing it dried out. But if you were to see them fresh, they are smaller cucumber seeds. They are actively moving. All right? And then you have to have basically a larval flea pick it up so that you could then start developing into the cysticercoid, which is the infective stage. Questions? Yep. Yes, they can. And that is partially why, I don't know, has anyone had a dog or a cat with bad tapeworms? But how do they treat them?
Well, with my just you didn't have to like have fleas, so it was just like the one you had, but they gave a shot.
They also treated for fleas too,
Oh, okay.
right?
I think so.
Yeah. So yeah, if you come in with tapeworm, they don't even check to see what kind of tapeworm. It's just the assumption. Okay, we're going to treat anti-helminthics and also treat for fleas. Same thing. All right. So human cases, yeah, you get it. Majority of them are kids. Why is that? Who knows? Who knows? Could be that adults are more resistant. Maybe our immune system's better at knocking it out and fighting the establishment of the infection. Or what I kind of think is the case, children are more likely to consume fleas. Not always. I mean, you see some adults that snuggle and bury their face in their animal's fur and definitely probably inhale fleas along the way. Definitely the kids. What type of pathology? Minimal. So, I say Tinea saginata. This is if you have an adult tapeworm. Again, just kind of minimal pathology. And here's an example of our egg packet with the eggs inside of it. And I think some of our slides you can see the packets. You can see the eggs that are in packets. You have to check. Any questions? There it is. All right. So that's going to wrap up test material. I thought I'd get through. So that was a good time. That's going to give you, the week to study, to prep for it, prep for the exam. Definitely, I'm going to say diagram out your life cycles, diagram it several times, try to get a feel for it. Maybe help yourself out with, just making lists of definitive hosts for the different parasites and larval hosts, kind of keeping track which ones are trematodes, which ones are monogenes, which ones are tapeworms, and so forth. If you can keep the larval stages, if you can keep track of the larval stages, that can help. That can help you with the exam. All right. Questions? Yep.
Review quiz for this.
That's a good question. I think I have one. It probably helps a little bit. Let's see here. Request my login.
And then, like the answer to the quiz will just be available sometime this week.
They go live tomorrow at like 8 in the morning. Let's check here. Quizzes. Yeah, I actually have two. Do you want them? So I've got one with just life cycles and one with diversity.
You just want to put them up with the answers.
We'll do the diversity one. Yeah, we'll do the diversity one as a real quiz. That's 15 minutes. Make available. All right, we're going to make that due on Tuesday night. So then Wednesday morning, the key will go live. Due date tomorrow, 11:30. I'm going to do that. We'll say after availability. Do that. So the diversity one still kind of gets you in reviewing everything. I may have Hymenolopus on there. So I may have Hymenolopus on there. I'm going to probably have to go back and look at that question. So any of them on Hymenolopus, I'll just have to take them out. So we'll adjust those grades. This life cycle one. Oops, hold on, let me go edit. Let me check the diversity. No, there's none for Hymenolopus. We're good on that. All right, so the other one, we will just make the cestodiversity, the life cycle one. We'll mark that as practice since we had a life cycle quiz here. So we'll just do that in place of it. I'm going to make it unlimited attempts. I don't know what the time. I had 15 minutes for the timer. So what you can do is just do it as many times as you want. I'm going to have it set up so that it's hidden. I can't see the results. But this way you can just kind of take it as many times as you want just to kind of practice. So that way that won't count. So those are up. Anything else? So we're going to start with the nematodes. The nematodes is, we're going to cover the nematodes and the acanthocephalans. So 2 groups, and then we'll get to the protozoans. So the nematodes, we're going to talk a few more life cycles, like invertebrate hosts and vertebrate hosts. We don't have, I'm going to say we don't have as many species to look at in the lab. That's why I don't think we need like 4 weeks between exams, but we will kind of whip through it a little bit. All right, I think we'll just end here for today. Start reviewing. Look over your notes. What we'll also do on Wednesday is if you have questions about the material, bring those questions to class. So if you have a question, somebody else may have the same question. Oh no. Darn it. I did not see any of those. Oh, sorry about the mic.